So it's a real pleasure to be here with you. And first, I'd like to thank the organizing committee for inviting me and presenting some of the applications that we've started developing with Stan in the context of human genetics. So I know most of you here probably are not in the field of human genetics. So what I want to do is to give a high-level overview as to what the state of the art is in human genetics and give some historical context as well as to how we got here. So a few things. First, rare diseases, as many of you may be aware, run in families. If you have cystic fibrosis, you can ask the question, what is the risk to your neighbor? And that answer is about 0.04%, matching the population prevalence. If you ask the question, if you have cystic fibrosis, what is the risk to your sibling? That answer is about 25%. And if you ask the question, if you have cystic fibrosis, what is the risk to your identical twin? And that answer is approximately 99%. These types of comparisons suggest that variation in your DNA influences disease risk. In the context of common diseases as well, you could do this type of comparison where you ask the same questions and you get some statistics that give you estimates as to whether or not the extent to which a disease is heritable. So in the context of type 2 diabetes, you can ask, if you have type 2 diabetes, what is the risk to your neighbor? And that answer is about approximately 10%, matching the population prevalence of type 2 diabetes. If you have type 2 diabetes and you ask the question, what is the risk to your sibling? That answer is approximately 30%. And for identical twins, that answer is more than 50%. So even in the context of common diseases, we have strong support just from familial comparisons of getting estimates of risk to disease. You can get some estimate of how heritable a disease is. So, for disease that we think of as a multifactorial disease like type 2 diabetes, you see strong evidence of risk in disease being driven by variation in DNA. So just to give you more of a historical context as to how the field of human genetics progressed over the past couple of decades, in the context of rare diseases, from 1980 to 2000s, the field was extremely successful in mapping genes that drive rare disease risk. However, for common diseases or complex traits, those 2 decades are what are considered the dark ages of human genetics. We had very, very limited success in being able to identify genes or mutations that drive some of that genetic risk. Drive some of that disease risk. In 2007, the HapMap project and other types of projects that started collecting information about genetic variation in the human genome was considered the breakthrough of the year. I think a lot of the technologies and a lot of researchers around the world gathered together and started characterizing where mutations in the genome exist. Now, many of you may be familiar, at least in the popular press, of the past decade or past 2 decades of genetics research and what's been called genome-wide association studies. From 2000 to today, that has been the primary engine of discovery for human genetics. So just to give you some background, just to give you a high-level overview as to what a genetic association study is, what you typically do is just collect a lot of individuals that don't have the disease. You label them as controls. You also collect research participants that have the disease. You consider them, quote-unquote, patients in your case control group. And then for a given genetic variant, assume that this is a single genetic variant, the reference allele is A, and then the mutation is C. You compare the frequency of that genetic variant in control, so it's a simple 2x2 table. And then if you see it overrepresented in patients, then you'd have strong evidence of it being a genetic variant that associates with the trait of interest. In this context, it being the disease. So in a typical genome-wide association study analysis, what's done is you apply logistic regression to this data. You adjust for some covariates that typically represent some form of membership across multiple populations. So you take the genetic data, you apply PCAs, and then you take those principal components as covariates in a typical genome-wide association study when you're trying to assess for evidence of association. Now, for the past 2 decades, this GWAS exercise has been quite fruitful in the sense that it's led to the identification of a lot of mutations or genetic variants across the genome that are associated to some trait that has been studied. However, as many of you may, actually I think Andrew Gellman would strongly state, or at least I've started gaining appreciation for this, the field has been dominated by null hypothesis testing. So basically all these quote unquote associations, the main objective is really to reject the null hypothesis. And once you start seeing the data emerging from when you go to large sample sizes, you start seeing the limitations of thinking about null hypothesis testing. So just to give you more background as to what a GWAS study data analysis typically looks like, when you assess for association across hundreds of thousands of genetic variants, you typically plot on the y-axis the minus log 10 p-value as strength of evidence for rejecting the null hypothesis. And on the x-axis is just a data point for a particular genetic variant that's located somewhere across the genome. This is what's called the Manhattan plot. And the reason it's called the Manhattan plot is that once you identify evidence of association, you see these sort of skyscrapers emerging. So you may ask yourself, why the asterisk up here? Well, the reason why I put this asterisk up there is that these GWAS studies, the bulk of them, have identified genetic variants that associate to disease. However, the majority of the genetic variants fall in this regime, where they are common genetic variants with very weak effects. So if you take the distribution of effect sizes for those genetic variants, the bulk of them fall with below 1.1, I would say. So that shows very strong limitations as to what we can come up with in terms of thinking about, what's been moved forward as personalized medicine or precision medicine. It's really hard to take that information about some genetic variants being associated with disease that only increases disease risk by less than 10%, for example. So in the context of type 2 diabetes, if you plot on the x-axis the minor allele frequency of genetic variants that have been associated to disease, and on the y-axis the odds ratios, you see that the bulk of them are very near 1. However, there's good news in that now as we're moving into these large-scale sequencing studies, we're starting to see signals emerging from these variant association studies that are now with stronger effect sizes. In fact, some of them are starting to show effects consistent with protection to disease. So if you're a pharmaceutical company, for example, you would be extremely excited by the property that there are mutations that break genes that protect individuals from having human diseases. So these experiments of nature that protect can guide selection of drug targets, and that's been, quote unquote, the hope of human genetics ever since the beginning, I think, for a lot of people at least. And the typical scenario is that you do a case control study or a population cohort study combine medical information about those individuals with genetic data, and identify mutations that lower risk for disease. Now, there are only a handful of examples of protective mutations that confer strong effects. I think the one that's the most successful to date that went from genetic discovery to passing phase 3 clinical trials, showing consistent effects in the therapeutic strategy with effects that you see in humans, is the example of PCSK9 for LDL and myocardial infarction. Now, the story there is that a sequencing study was done in African Americans where they identified genetic mutations that completely break the function of this gene called PCSK9, reduce LDL levels by like about 80% from the population mean. So I think you'd see LDL levels around 20 in these individuals that carry those loss of function mutations, and lowers myocardial infarction disease risk for those individuals like about 50%. I've participated in some studies where we've started identifying these protective rare variant mutations with strong effects in the context of inflammatory bowel disease. So in Crohn's disease and ulcerative colitis, we identified a mutation that breaks this gene called CARD9 that confers 4-fold protection against Crohn's disease and ulcerative colitis. Similar story with RNF186, where we identified a mutation that confers 2-fold protection against ulcerative colitis. And some of those stories just came one by one in some diseases. However, it hasn't been the great sleuth of findings that you identify with common variants. And the reason, of course, is that these are rare genetic variants. And therefore, you'll probably need a lot more samples in order to get to a point where you're getting some precise estimates accurate estimates of effects. So you may ask yourself, well, why the need for STAN in human genetics? And this is something that I quickly realized after participating in a lot of these genetic studies, is that we needed to rethink about the way we think of statistical methods in the context of human genetics. So parameters become more exciting and informative. Concepts like location, scale, correlation of genetic effects become quite what you want to end up communicating to the public, as opposed to identifying genetic variants that have some non-zero effects or rejecting the null hypothesis. And p-value is not informative, especially when you realize that the entire genome is associated to disease. So, when you get to a point of collecting very, very large sample sizes and you do genetic studies, especially if you apply logistic regression, all your p-values look like times 10 to order of times 10 to minus 100, times 10 to minus 200, times 10 to minus 300. It just doesn't mean anything anymore. So just to give you a bit more background, in the age of common variants, we were used to looking at genetic variants that were sparsely separated across the genome. So these are genes, so a couple of genes in the genome, and this is the entire tract of the genome. And typically you'd have a few common genetic variants that you then move forward in a genetic association study. Now, there are new technologies that are transforming biomedicine. One of those technologies is, of course, DNA sequencing technology. So many of you may have seen this plot in some of the popular press. What you're starting to see is the drop in cost of sequencing has plummeted over the past 15 years. Think, in 2001, the cost of sequencing a human genome was approximately $100 million. And today it's it's affordable, at least in the research setting. You can sequence a whole genome for about $1,000. You can sequence a whole exome for approximately hundreds of dollars. And you could do whole genome genotyping for approximately $30. And I can describe that later if you're. So in the age of rare variants, once you move to sequencing, what you end up getting then, especially as you sequence very large numbers of samples, is you start coming across this picture, where you have genetic variants densely represented almost across the entire genome. So you'd have, if you sequence 1 million individuals, you'd almost guaranteed to have every site in the genome mutated in at least one individual. Except that it's embryonically lethal, of course. So in the context of inflammatory bowel disease, when you look at the data and you sequence 100,000 individuals, you go from 3 variants represented in the gene to almost every site in the gene having a mutation in the populations that we've sequenced. So just to recap, in a single gene, when you sequence over hundreds of thousands of humans, you get over 1,000 genetic variants. In comparison, in the age where you didn't have DNA sequencing as a technology, you are only allowed to see the data from 3 common variants. These genetic variants or mutations have predicted functional consequences. For example, some of the mutations that you see, you can predict just in silico by taking the genetic code and predict that it's supposed to have a silent or a very benign effect on gene function. Similarly, you can take the genetic code and predict that it'll change the amino acid of the protein at a particular location. In addition, you can also predict that there are some mutations that lead to loss of function effects, meaning that it completely knocks down the function of the gene. Just to give you a sense of the data as well, the data comes from multiple populations. In a lot of the studies that we participate in, we have genetic data, genome sequencing data in the context of multiple diseases for Ashkenazi Jewish population, Finland, French Canadian, African American, Latinos, non-Finnish European, and the landscape of genetic variation looks quite different across populations. So just to give you a flavor as to how the data from genetic study typically looks like, if you are involved in all of the projects, you'd be able to get access to individual level data. The main challenge is that most researchers actually don't have access to each of the project's individual level data. So at the end of the day, what you end up working with looks like as follows. It looks like summary statistics. So somebody has gone ahead and applied some logistic regression analysis to the data. And for each genetic variant in the genome, they give you some reported effect size estimate along with some standard error for that effect size. You can obtain annotation or predicted functional consequence of that particular mutation. You know about the gene and the population that it came from. So this lends itself nicely to a lot of the more advanced statistical modeling that Stan affords. In fact, the package BRMS facilitates multi-level modeling with meta-analysis type data, and this is something that we've been playing quite a bit in my lab. The idea is that if you do have access to this type of summary level data, you can start taking these beta hats and standard errors as inputs and doing regression analysis, multi-level modeling, using the gene as the group term, the grouping, and the annotation as the group level effects. And then you have some population level effects represented as well that you'd probably want to assess for to get estimates of parameters. In this simple context where we're looking at 3 genes, you can see for variants that have predicted loss of function effects, if you have this multi-level model, for each of the genes, you can get some posterior estimate of the effect size for those types of variants. Similarly, for missense variants, and in addition to that, you get additional parameters from BRMS that tells you a bit more about the standard deviation of effect sizes across these group level effects. The other thing that we've been doing that's a bit more mature than the BRMS modeling activity, in the BRMS modeling activity, what we're excited about is the potential for multi-level modeling to ask different questions about and to obtain parameters across, for example, correlation of genetic effects across populations, correlation of genetic effects across different classes of variants, and correlation of genetic effects across genes, for example. We're also interested in scale and location of genetic effects. So if you're interested in identifying genetic variants that confer protection to disease, maybe for a particular rare variant you don't have enough information, but a particular gene you probably have support for it. Anyways, so to move forward, in the context that's a bit more mature is the analysis that we've done with population cohorts, where we've started to estimate parameters across multiple phenotypes, where our main interest is to estimate the extent to which we see correlation of genetic effects across multiple diseases, for example. So just to give you a big background of this data set, the UK Biobank data has approximately 500,000 participants. It's a population biobank cohort that's available to any bona fide researcher in the world. About 330,000 individuals of the 500,000 data set are European British. It has a large numbers of phenotypes and genetic data. So some of that phenotype comes from hospital inpatient record data. Some of the phenotype comes from verbal questionnaires, so just asking an individual whether or not you have a particular disease. Some of that data comes from cancer and death registry data sets, sources. They have imaging, biomarker, and lifestyle measures as well. So in the lab, we've started to use STAN to estimate genetic parameters using those summary statistic data inputs. For example, here is just an example of what that data looks like. So for phenotype 1, for any particular genetic variant, you get beta and standard error from the logistic regression analysis that was performed by somebody else. Similarly, for phenotype 2, you get the same types of statistics, summary statistics. So you use that data as input, and what we've done is to develop a mixture model, multivariate normal mixture model with two components, where the first component is just the point mass at 0, and the second component is a multivariate normal centered at 0 with some unknown variance covariance matrix that we're interested in estimating. Similarly, we have parameters like pi. We're interested in the membership of those genetic variants across those two components. We're interested in tau, or the spread, the scale of effects, and omega, which is the genetic correlation matrix that we're interested in estimating. This research activity has been led by Chris DeBover, who is in the audience, and he presented a poster yesterday. I hope that you guys had the chance to meet with him. So just to go back, how is it that we've used STAN? We've used STAN to fit the summary level data to this mixture model. One of the applications that we've moved forward with is asking, given that you do have access to hospital record data, so you may have some disease phenotypes that are probably a bit cleaner coming from physicians, and you also have access to disease phenotype labeling coming from verbal questionnaire data, so just ask an individual whether or not they have a disease. We can start using this mixture model to assess whether or not the genetic architecture of those two ways of phenotyping are at least consistent with each other. So the good news is that for most diseases, the case is that the correlation of genetic effects is pretty consistent. However, you do have diseases where the correlation of genetic effects is quite less than 1. For example, migraine is one of those examples where the correlation of genetic effects is around 0.5, which is probably consistent with the property that you do have a lot of individuals that are reporting as having migraine that probably have some form of other phenotype that gets confused for migraine. So with STAN, one of the nice properties that from reading the manuals is being able to get guidance as to how we should interpret the MCMC runs when we do the pairwise comparisons. And because there are a very large number of pairwise comparisons, and this is quite a large data set, even at the summary level scale, having some guidance as to when the sampling has converged is quite informative. So the R-hat distribution, at least for the genetic correlation parameter for these comparisons that I showed in the plots, looks to be pretty much centered around, around 1. So we've started building tools in the lab and making some of these inferences publicly available to the world. In this context, we've started providing estimates of these genetic parameters, like correlation of genetic effects across diseases, in this app called biobankengine.stanford.edu/gcore. And here you can start seeing which diseases seem to have shared genetic effects. The really neat thing about human genetics, at least right now, is that we're starting to see this picture emerging across many of traits, if not all the traits, where almost the entire genome plays a role. In terms of having very tiny effects. Now, those rare variants that have strong effects, I think those are the ones probably that give you a bit more of a guidance as to specific genes that you may want to go after. And that's been a bit more of a challenge, and I think that's going to be a challenge for this generation of geneticists to think about how you can take these very dense set of genetic variants and make better inferences from the data, as opposed to doing this p-value style association analysis that had been done traditionally. So there's another project that I do want to share with you, and it's similar flavor where we do have a mixture model. Instead of looking at 2 phenotypes, here the comparison is looking at the genetics of a disease or a particular trait across males and females, and asking the question and evaluating how much of those genetic effects are shared across males and females, and identifying when is it that those genetic effects are either specific to males or females. So this research has been led by Emily Flynn, who's a PhD student in the department. The nice thing about working with Emily is that in a class I taught some of the activities from the STAN manual, and she was able to pick up STAN quite quickly and be able to come up with a nice mixture model, apply some of the advanced techniques to the human genetics data, and come up with inferences that I think, in the course of 2 weeks, if you asked me whether or not I could come up with those types of inferences 10 years ago when I was the same age as her, the answer is clearly no. So, much credit to the STAN developers for making it easy as possible for anybody actually to start picking up STAN and applying it to some of the very exciting data sets that are emerging from different fields. So one of the nice things that she's able to do is to take these genetic correlation estimates using this 2-component mixture model and compare it to the literature. And for most cases, you see pretty consistent estimates of correlation of genetic effects across what she gets from the UK Biobank data, which is in red, and what's reported in the literature. In fact, she's also able to pick out that for waist-hip ratio, when you compare the genetics of waist-hip ratio in males and females, you don't see the patterns of correlation of genetic effects that you see for other traits that have been reported in the literature as well, matching what we see in the literature. So not only does she focus on those traits that have been reported in the literature, where they've done the comparisons of the genetic architecture across both males and females, she's also able to take this very rich phenotyping data from the population biobank and also estimate these parameters across more than those traits, and identify sets of traits where the correlation of genetic effects is less than what you'd probably anticipate to see. So the cool thing is that for most traits, you're seeing pretty consistent, you're seeing pretty high correlation of genetic effects in males and females, and there are some exceptions, but those exceptions are quite interesting as well to move forward with. Now, the final mixture model that I want to share with you, that it's an application of STAN in the context of human genetics that we've been working on, is the context of is also led by Emily Flynn, where she sets up a mixture model where she's fitting a 4-component mixture model. First is the point mass at 0. Second is just a female-specific effect model, a male-specific effect model, and effects in both sexes. And the nice thing is that she's able to get parameter estimates of how many genetic variants across the genome fall in each of those components and identify sex-specific effects. So if you were to do this in the GWAS, traditional GWAS setting, what you would do is to plot the Manhattan plots with the minus log 10 p-values on the y-axis, the genetic variants on the x-axis, and maybe one way of doing the comparison is by looking at the minus log 10 p-values, but as all of you know, that's not a kosher way of doing it. In fact, I still think that this null component here does bring in some issues, which is something that we're still trying to handle. So with that said, though, we are able to identify genetic variants that have sex-specific effects. So here are sets of genetic variants, a handful of genetic variants that, for height, have an effect, if you plot it in the traditional sense in GWAS land, have evidence of association with height, and in females, you don't have the association. So just to get a tally as to how many sex-specific genetic effects we identify across the genome in this very large cohort, for waist-hip ratio, like fat ratio and arm fat ratio, we've identified over 50 genetic variants that have sex-specific effects. And in the context of males, we've only identified 4, but it's quite exciting then to move this forward into to share that with some of our biologist friends. So just to conclude, I think I have 5 minutes left. There's ample opportunities in linking medical population data, human genetic data, and I think STAN has been an extremely important platform, at least for us in our lab, to move forward some of our research activities. And one thing to take away, if you take away anything from this talk, is that, at least in the context of human genetics, you clearly see the opportunity that exists in moving away from thinking about frequencies hypothesis testing. And I'd encourage you as a community to get a bit more involved in human genetics as well, if you can. All right, thank you.
Hello. Questions? Oh, all right, sorry. Going back here? Let's go from, there we go.
Thanks, very interesting talk. I didn't quite understand why you're using a mixture model to have the different sex effects rather than having one sort of sex beta and then which way it goes, it's male or female.
Yeah, great question. So, the reason is that we're actually quite interested in getting an estimate of the parameter pi. So we do want to get, at the end of the day, a proportion estimate as to how many things seem to be, strongly supportive of it being consistent with no effects across both sexes, and then having an estimate across the genome how many seems to be sex-specific. And the reason that we moved in this direction is that we approached it with the premise that actually a large fraction of the genome has some non-zero effect. So that's the reason why the parameter pi actually is to us at least more exciting at this moment in time. Nonetheless, if you were interested then in just looking at a specific genetic variant, then moving with that approach would also work.
Questions? You're up.
If your hypothesis is that there are many variants that have small effects, wouldn't that lend you to want to use this sort of model where you can allow for small effects in either direction rather than saying that there's no effect, no male effect?
That's right. So yeah, that's right. So after hearing Aki's talk, actually, I think that's where the having a model where you just completely take out the null component would make sense. Initially when we approached this, we thought that it would make a lot of sense with the way the human genetics community moved to communicate that inference of the null component, right? Realistically, if I came to the human genetics community and told them, the null component doesn't exist, they would push back quite a bit. So it's pragmatic, it's a practical issue, but realistically, I think that the next steps for us is to completely remove that null component, and start using some of the techniques that Aki has been working on.
All right, we'll go over here, and then Eric.
Thank you for your talk. I was wondering what cutoff you use to decide that there is an effect, and how do you sort of sell this cutoff, for example, to grant agencies that might be more used to seeing a p-value? How do you sort of sell that?
Yeah, great question. So you start moving away from cutoffs to talking about posteriors, right? Posterior probabilities as well. So if you are talking about mixture model with multiple components, then you talk about posterior probabilities of the genetic variants belonging to either of the components. I'd completely agree with you that, or at least I'd acknowledge that grant agencies, at least that have been used to funding genetic studies, would have a very difficult time in moving from p-value cutoffs as a rule to posterior probabilities, especially when you're talking about distributions with specific parameters, right? Or even if you move away from mixture models, how you start thinking about posteriors of effect sizes, right? And I think that's going to be the challenge in communication for us. I think that the observation that the genetics community is seeing right now is that, if you plot those Manhattan plots, the entire genome is spiked up. And that's what you'd actually anticipate if you listen to Fisher, which is kind of weird to bring it into a Bayesian context, but actually that's I think that's going to be part of the communication challenge that the genetics community is going to come across. The nice thing is that we do have strong data to suggest that actually moving away from those p-value thresholds is probably the right way.
So a lot of people that I talk to that do this kind of work, they use a lot of approximate methods. Are you guys using full Bayes for this? And what's your experience with computational aspect of it?
Great question. The computation side has been a bit of a challenge in going and in adopting Stan for everything that we do in our lab. So if we did full Bayes on this, how many days would it take, Chris? Okay, so it'll take quite a bit, and it'll take a lot of computational resources. The optimizing function was quite nice in at least being able to assess when things were working or not, and how it would look across multiple traits. The challenge with optimizing, as Michael Betancourt told me when we met, is that it'll give you inferences that are completely wrong. And we've, exactly. So we've seen that, and we've therefore for a lot of the activities that we've moved forward, we've asked targeted questions. So at this moment in time, we still can't look at thousands of phenotypes and do pairwise calculations across them. Nonetheless, I think that's a challenge that exists right now for Stan, and we'd love to be able to even provide some data that you guys can play around with to assess whether or not there are some computational strategies that you can move forward with.
Hi.
Yes.
I was wondering if your lab has approached the problem of people who have mutations but do not display any symptoms or any phenotype? How do you incorporate that information? Because it's something like a counterfactual that, people have these mutations, but they never develop the disease. So when I see these studies where they only do case control, we completely ignore that part of the problem. And that's what always kind of gives
Yeah, that's right. So, one of the cool things that's emerging from the common variant world, at least, is that, those tiny effects do add up additively. Therefore, for most individuals, we fall along, for all of us, we fall somewhere in the distribution in terms of risk to disease. And sometimes when you observe mutations that have strong effect on disease risk, for example, assume that you're thinking about breast cancer, and assume that you see a strong effect mutation in BRCA1, right? But you don't observe the individual to have disease. Sometimes that disease risk is modified by the combination of all these really tiny effects. So there are modifying contribution of genetic effects in the genome that would modify the effect that you'd probably anticipate to see in an individual that has disease. So I think this right now we're at a moment in time where because we have genome sequencing data and because we have been able to estimate effects, those tiny effects across the genome, we can now start to bring them together to ask more targeted questions about individuals that are probably not breast cancer cases, are not cases, but probably have some mild form of disease, and individuals that probably don't have the disease at all, right?
Anybody else? Another question? You guys are questioned out. So thank the speaker.
Thank you.
Thank you very much.